Welcome to this video where I'm going to be talking about nuclease assays and DNA denaturing gels. So to start out, we need to make sure that we have all of our tubes labeled with our different proteins, and then we're going to load in a DNA solution into a plate so that we can add in the proteins after that. Make sure that you keep your DNA on ice because it'll just keep it more stable. It's not completely necessary, but good practice to do. So after you get all the DNA and the proteins in the correct concentrations, we're going to go ahead and transfer those over to a plate so that we can put that into a thermocycler. For this experiment, we're going to be using 1 micromolar of each of our proteins and a DNA concentration of 50 nanomolar for the final solution. Okay, now we have our plate ready to go, and we're going to start labeling it so that we have different groups for our DNA and our proteins. And then what I'm going to do is go ahead and load in the DNA first. So that's all ready to go. We have 2 different types of DNA in here, one with a epigenetic marker on it and one that's just a regular set of DNA. And then what I'm going to do is go ahead and load in some proteins down below. And you'll see in just a second why I do this, but I want to be able to use a multi-channel so that I can transfer all the proteins pretty much at the same time. So here we go right here, transferring the protein in, and then at this point we'll have protein mixed with DNA. And now we can take our plate over to the thermocycler, and we're going to use this to heat up our reaction and make sure it stays constant during the reaction. So when we load it in, we're going to start up the thermocycler, and then we're going to actually create a custom protocol. So what we'll go in and do is edit an existing protocol, and we're going to add a step at 37C for 60 minutes because we just want to see products for this reaction. And then our quench is going to be at 50C, and that's going to happen for about 30 minutes. And then we put a final step of 4C so that it can hold there and stay stable afterwards. So while that's running, we're going to go ahead and make our quench buffer. And the quench buffer is made up of 2 different things. We have 0.5% SDS and we have proteinase K. In addition, we're going to go ahead and make the DNA denaturing gel. So this consists of 7.95 molar urea, 21.7% acrylamide, and 1.14x TBE. So we're going to go ahead and mix this together, and I always put the urea in first because it needs to dissolve. So we'll go ahead and do that. I tend to put a cap on top of this and then shake it up and down so that it goes into solution. And then I also will sometimes grab it in my hands to warm it up a little bit. And then after we get that done, we're going to add formamide to it. So formamide should really be done in the hood. So I went ahead and put 5.5 mils of that into the test tube and then added 40 mils of our denaturing gel solution. So you can see here that I'm putting together the gel box and screwing that in. And the first thing we're going to do for this is check for leaks. So I pour isopropanol in there and just make sure it's looking all good. And now we're going to actually add in the 220 microliters of TMED and then 22 microliters of APS. Once that sets up, we're going to let that go for about 1.5 hours to make sure that it polymerizes completely. And then we're going to go ahead and store it in the paper towels overnight. And what you're going to do is wrap it up in paper towels and then add a little bit of buffer solution to keep it moist. And then you can go ahead and put it into a refrigerator. So once the reaction's gone for the amount of time that you want it to go for here was about an hour, we're going to add the quench buffer. So what I'm doing here is distributing out 10 microliters of quench buffer into each of these wells so that I can use a multi-channel pipette and pipette 5 microliters into all of the reaction samples. So real quick, we're just removing the tops of the tubes, and then we're going to go ahead and add in those 5 microliters of quench buffer. Once the buffer's loaded, we're going to let this run for 30 minutes at 50 degrees C, and that's going to complete the quenching. Okay, after the quenching step has finished for our reaction, we're going to go ahead and place this in the -20 to sit overnight. And then the next morning, we're going to come unwrap the gel and load it into the gel box. You can see that we tried to get it really tight in there. And then we're going to start making the buffers. So we have 0.5x TBE and 1x TBE. So the 0.5 goes into the top chamber and the 1x goes into the bottom chamber. And then we're going to fill a syringe and actually clean out each of those wells individually because the urea tends to stick around and we want to get that out of there as possible. Now we're going to put it into an incubator so that we have a constant temperature of 40 C. And then after that runs for about 2 hours, we're going to clean out the wells again. To prepare the samples for loading into the gel, we're going to take 7.8 microliters of the reaction solution and mix that with 3 microliters of formamide that has EDTA in it. And then we're going to add 1.4 microliters of 1x TBE, and then we're going to let that sit at 95 C for about 10 minutes before we add in 4 microliters of 40% sucrose before spinning down and then getting ready to load into the gel. And then we can go ahead and load in our sample. So here I'm adding 13 microliters of each of the samples into the individual wells. And once that's loaded, we're going to let that run for 6 hours, and then we can remove the gel from the gel box. So we unload it and then we take off the ends, and now we can take the plate and put that into a plate reader. Right here we have a Typhoon that we are going to work with, and it's going to scan our gel and tell us exactly where our bands are. So we do a pre-scan first, and that's so that we can target where we want to look and get really precise measurements. And then once that's done, you can take those results and import them into ImageJ and get peak integrations. And that wraps up running through a nuclease assay and taking it through a denaturing gel.